normal cell communication, there are three steps: reception, transduction, and response. Reception is the binding between a ligand, which is a signal, and a receptor, which is highly specific. There are three different sites they can bind to. G-protein coupled receptors work with the G-protein. Once the ligand attaches to the receptor, it will activate the G-protein and turn GDP into GTP. This will activate an enzyme called adenylyl cyclase, creating CAMP. When a signal is received, the tyrosine kinase receptor regions attach phosphates to the tyrosine can trigger multiple translected pathways at once. Each tyrosine picks up phosphates and it gets passed on to activate the next protein. When a signal is received, the closed gates open, allowing specific ions to enter. Once the receptor proteins receive the signal, the receptor will have to change shape and transmit signals. Transduction is a multi-step pathway that triggers amplification and produces a larger response. The most common messengers are cyclic AMP and calcium ions. CAMP activates a phosphorylation cascade, leading to amplification. Calcium ions are important because cells can regulate their concentration, and the more ions that get released, the bigger the response. The signal transduction pathway leads to the regulation of cellular functions. Some may regulate synthesis of or activity of enzymes, or affect the cell's characteristics. Epilepsy is associated with repeated and unprovoked seizures that occur due to a variety of factors. Epileptic seizures occur through local signaling when sudden and excessive electrical bursts in the brain fire between neurons. This can cause neural loss, behavioral changes, and irreversible loss of function. Picture two neurons and their parts. The process begins at the axon terminal buds of neuron A. Neurotransmitters or ligands are released to the synaptic gap and bind to a receptor on neuron B. Excitatory signals tell the receptors to open up ion channels and relay electrical messages, while inhibitory do the opposite. During a seizure, a cluster of neurons send an excessive amount of signals due to too much excitation and little inhibition. Glutamate is the main excitatory neurotranslator, and after reception, it opens ion channels to allow both positive calcium and sodium ions in. Incoming ions into the channels allow for depolarization, which makes the neuron more susceptible to fire a nerve impulse. Normally, inhibitors such as GABA tell the cell to stop the impulse from being performed. The excessive amount of glutamate in epilepsy acts as a ghost signal for firing action potential, causing excessive electrical bursts resulting in seizures.